or if you yourself are going to be applying to graduate school and are interested in knowing more about Cornell, we have a diversity recruitment weekend coming up in spring, and the deadline is in December. So the website is here, inclusivecornell.org, and there will be more information there for those who are interested. Okay, thanks, Matt, for setting up the talk so nicely. I guess my job here is to give the talk on the not so sunny side about species delimitation and whether it works or not. And I'm going to do that in the context of discussing a situation where we have evidence of speciation in the face of gene flow, so divergence with gene flow, and asking questions about how potential sampling biases might creep into the way that we use delimitation methods. Okay? So we all know that species don't come out of nowhere. They don't all of a sudden appear instantaneously, and that what this means is that species as entities actually have histories, and those histories include a number of different evolutionary forces that occur over variable time frames, and that's not only what makes species interesting, that is what makes species interesting, but it also means that in many cases we may have difficulty finding sort of generality in terms of the history of the species and what that means in terms of our ability to discern limits in those particular groups. So if you think about this, if we think about delimitation as a process in which we're actually trying to incorporate that history in our efforts to delimit species, and then we think about how we go about doing that, we have a second problem. And that is, on top of this variable history that exists among species, we impose yet one more non-random process, and that is how we sample in trying to delimit species, how we sample populations in the field. So what I want to do today is use endesiops, the mayfly that Matt already described, and use an empirical data set for that species complex, and ask questions about how, given that we know that there's divergence with gene flow in one of the species, in one of the morphospecies and one of the complexes, and I want to ask about how sampling on top of that underlying population structure confounds or not delimitation efforts. Okay, so our goal, as I just said, was to investigate the biases that may be imposed by population sampling in this situation, and I would argue, if we take from Matt's presumed benefit slides of delimitation methods, that objectivity and identification of cryptic species, that both of those benefits are actually moot if somehow the species delimitation methods don't perform predictably with something as simple as differential sampling. Okay, so if every person samples differently and every person is going to get a different result in terms of delimitation, then clearly there's an issue with the process itself. Okay, so I used endesiops, a species complex of mayflies that occurs in montane streams in Ecuador. Streams in mountains are obviously great systems in which to look at differentiation. Mountains themselves, especially in the tropics, offer a relatively large gradient in terms of climatic diversification. We expect some diversification of organisms in those environments. And we know a fair bit about, in this particular case, about the population structure of this species. So I chose one drainage, the Papayacta drainage in Ecuador, to analyze the situation further. So I should say, at this point, there is a single species of endesiops known from these streams. Our systematist friends, insect systematist friends, know for sure, they tell us, we know that there's differentiation in there. It's a complex, we don't know really where one species begins and one species ends, at least at this point. Okay, so this is the drainage we chose. Matt already showed you this. There's a fair amount of structure. These are just structure plots, population structure plots from DE-RAD data. We also know, as Matt showed you, that if we look at the preferred number of genetic genes that show up in structure, that there's probably a gradient going from species blue to species red here, with some admixture in between as you move up the stream here. And even though it's messy, we also have a significant pattern if you look across all populations of isolation by distance. Okay, so there are a number of things going on here in terms of population structure. The question is, can we actually say anything about where this species red with admixture in the middle. Okay, so in other words, we chose only individuals from parental populations that were not admixed, sort of forcing this species at this end of the continuum to be 100% blue, and those non-admixed red species. And then we chose individuals along the continuum of admixture that maximize the degree of admixture in the middle. So this is like, imagine the perfect situation. Your two species at each end of this transect are as clean as they can be in terms of genetic signal, and these guys in the middle are as admixed as they can be, okay? And then we imposed sampling scenarios by dropping individual populations from the transect, from the admixed individuals, from the admixed populations, and dropping more and more. So we started by dropping a single admixed population and ended by dropping the 5 all admixed populations, okay? So the question is, how do species delimitation efforts reconstruct the number of species in each of these cases? We would predict, if species delimitation are going to be useful, that independent of how you sample as a biologist, that you should get at least a similar answer. And here's what we found. So here are the different scenarios of sampling. And right here we have base factors and the 4 different sampling scenarios, okay? And so what you see is, as you predict, as you move from a continuous sampling situation to admixed populations not sampled, you get higher support for 2 species over 1 across this transect. Okay, so in other words, what's happening here is by not sampling admixed populations, you're inflating the support for 2 species over the same procedure applied to continuous distribution. Okay, so again, sampling in this case does seem to matter. In fact, all of the models showed favored 2 species over 1. However, there were fairly large difference in terms of the level of support for the different scenarios. In the second run was a little bit different, and in fact, chosen to represent sort of more messy data, okay, so more random data. And in this case, we didn't sample any individuals from parental populations or admixed populations in a selective way, okay? So individuals who were selected randomly to represent parental populations and admixed individuals in the middle. So what this means is in some cases, you have individuals sampled in what should be a pure species blue population that actually had a fair amount of genetic membership to the other gene, okay? Again, the same sampling scenario was imposed, dropping 1, 3, and 5 populations. And the results are exactly the opposite in this case, okay? So in this case, what happens is you actually have higher support for 2 species in the case of continuous sampling, and lower support for two species. Although all results support 2 species, you do have lower support for 2 species when you miss sampling the admixed populations. So I guess the question here is, if we are going to accept species delimitation methods as actually telling us something that's meaningful, the question is, can we live with this kind of variation in terms of the result that's based on purely whether you have sampled a population or not? And remember, of course, that when you're sampling populations up and down the stream, you don't actually know where the admixed populations are. At least most of us don't know a priori. And so this makes the sampling effort even a little more complicated, because it's not like you can target particular groups or particular populations that you know would enhance your probability of accurately eliminating species. Okay, so the take-homes are that sampling actually matters. It's something that we need to pay attention to, especially in situations where you might have some underlying population structure. I don't see a lot of potential for solving this at some sort of general level across species, because every species is going to be different in terms of the distribution or the underlying population structure. So it's not like we can, I don't think, at least at this point, come up with some threshold or number of, for example, the degree of admixture or gene flow above or below which we should or should not apply these methods. So that's a problem. We found high variance across relatively small changes in admixture across these populations. And so I think the bottom line is that we need to test the effects of all these different processes, some of which are intrinsic to species and some of which are extrinsic, but we don't know at this point whether species delimitation or how much species delimitation methods are affected not only by admixture and sampling, but by other processes such as selection, sample sizes, allele frequencies that the snails that you're using, and so forth. Okay? So until we have tests of that, I would argue that species delimited based on genetic data alone should be taken with a grain of salt. Until we have further corroboration with different kinds of data and better assessment of how the models are performing, I think we should proceed with caution. So that's what I have. I want to thank everybody, especially the organizers. Happy to